Mechanisms of action of phytochromes. CSIR Net JRF Life Sciences. Paragraph format. Maximum word limit. No tables based on previous year trends. Exam oriented and detailed. Phytochromes are a class of light-sensitive proteins found in plants, fungi, and some bacteria, playing a crucial role in the regulation of light-dependent developmental processes. They mediate a variety of physiological responses to red R and far red FR light, thereby influencing processes like seed germination, photoperiodism, shade avoidance, and flowering time. Phytochromes are part of the broader field of sensory photobiology, which deals with how organisms detect and respond to light signals. Understanding the structure, function, and mechanisms of action of phytochromes is essential for unraveling their role in plant biology. Structure of phytochromes. Phytochromes are generic proteins composed of two identical subunits, each of which consists of two main domains, the chromophore binding domain and the kinase-like domain. The chromophore is a tetrachiral structure derived from the liver and its derivatives and is responsible for the light-sensing capabilities of the phytochrome. This chromophore undergoes a photochemical transformation upon exposure to red or far red light, switching between two interconvertible states, PR phytochrome red and PFR phytochrome far red. The PR form absorbs red light approximately 660 nanometers, while the PFR form absorbs far red light approximately 730 nanometers. This conversion is central to phytochrome signaling. The kinase-like domain in phytochromes is involved in transmitting the light signal into a biochemical response within the plant. It is often associated with signaling pathways that regulate gene expression and physiological responses. The phytochrome family has different isoforms, each of which has slightly different functions depending on the plant species and environmental context. Function of phytochromes. Phytochromes play a central role in the regulation of light-dependent processes, responding to both the intensity and quality of light. They can sense changes in the light environment and mediate adaptive responses to optimize plant growth. Some of the major functions of phytochromes include seed germination. Phytochromes regulate seed germination by detecting the ratio of red to far red light. In many plants, the presence of red light promotes germination, while far red light inhibits it. This light perception ensures that seeds germinate in optimal light conditions, particularly under conditions where the canopy might block red light and increase the relative amount of far red light. Photoperiodism. Phytochromes are critical in regulating photoperiodic flowering, which refers to the plant's ability to flower based on the length of day and night. The conversion between PR and PFR states of phytochromes helps plants assess day length, thereby regulating the flowering time. Short day plants require a certain period of uninterrupted darkness to flower, while long day plants require extended daylight periods. Shade avoidance. In natural environments, plants growing in competition with others may experience reduced light levels, particularly in the red and blue wavelengths, due to shading by taller plants. Phytochromes help plants detect the shade and activate a shade avoidance response, which includes elongating stems, leaves, and petioles to improve light capture. Phytochrome signaling is involved in sensing the far red light enrichment typically caused by shading. Circadian rhythms. Phytochromes play a role in regulating the circadian clock of plants, which governs daily physiological processes like somatal opening, photosynthesis, and flowering. Phytochrome signaling influences the expression of clock genes, ensuring the plant's internal rhythm aligns with the external light-dark cycle. Mechanisms of action of phytochromes. The action of phytochromes begins with the absorption of light by the chromophore. The conversion of the PR state to the PFR state, triggered by red light, is the first key step in the signaling cascade. The PFR form is biologically active and binds to various phytochrome interacting factors, PIS, a group of transcription factors that mediate the downstream effects of phytochrome signaling. Upon light activation, the PFR form of phytochrome interacts with PIS in the nucleus. In the absence of light, PIS are typically active and repress the expression of light-responsive genes. However, when phytochrome is activated by red light and converted to PFR, it causes the degradation of PIS, thereby relieving the repression on these genes. This leads to the activation of gene expression related to photomorphogenesis, seedling growth, flowering, and stress responses. Phytochromes can also modulate the expression of phytochrome-regulated genes by interacting with other signaling pathways, such as those mediated by auxins, gibberellins, and cytokinins, integrating light signals with hormonal regulation. For example, phytochrome-induced degradation of PIS can activate the transcription of genes involved in cell elongation and differentiation, which are essential for photomorphogenic responses. Another key aspect of phytochrome action involves the feedback regulation of light signal transduction pathways. Phytochrome signaling not only activates light-responsive genes but also helps reset the plant's response to light. This involves complex feedback loops that fine-tune the plant's responses to environmental light changes. Role of phytochrome in plant growth and development photomorphogenesis. Phytochrome signaling plays a major role in photomorphogenesis, which is the development of plants in response to light. This includes the regulation of seedling detylation, where dark-grown seedlings, upon exposure to light, undergo rapid changes such as chloroplast development, leaf expansion, and stem elongation. These changes are crucial for optimizing the plant's ability to photosynthesize and grow under light conditions. Stomatal regulation. Phytochromes also regulate stomatal movement, which is essential for gas exchange and water regulation. In response to light, phytochromes can influence the opening and closing of stomata, facilitating photosynthesis while minimizing water loss, especially during periods of high light intensity. Previous year CSIR net questions. CSIR net June 2021. What is the primary light absorbing chromophore in phytochromes? Answer. The chromophore is the liverin, which undergoes a photochemical transformation to facilitate the conversion between PR and PFR forms. CSIR net 2019. Which form of phytochrome is responsible for activating the signal transduction cascade? Answer. The PFR phytochrome far red form is responsible for activating the signaling pathway and mediating light responses. CSIR net June 2018. Phytochrome-mediated shade avoidance responses are primarily regulated by which class of proteins? Answer. PIS phytochrome interacting factors are key regulators of the shade avoidance response in plants. Conclusion. Phytochromes are central to regulating the plant's ability to perceive and respond to light, influencing a broad range of processes essential for survival and adaptation. Their structural properties, including the chromophore and kinase-like domain, allow them to function as light sensors, while their signaling mechanisms activate or repress key growth and development genes. Understanding the molecular action of phytochromes provides insights into how plants optimize their growth in changing light environments, thus contributing to enhanced productivity and adaptability. Would you like to proceed with another section or topic in sensory photobiology? You said, cryptochromes. ChatGPT said. Cryptochromes, structure, function, and mechanisms of action. Cryptochromes are a family of blue light sensitive proteins found in plants, animals, and microorganisms, playing a pivotal role in the regulation of light-dependent processes such as circadian rhythms, photomorphogenesis, and flowering. These proteins are part of the broader category of photoreceptors, which detect and respond to light signals, influencing various physiological processes. In plants, cryptochromes interact with other photoreceptors such as phytochromes to modulate growth and development in response to environmental light conditions. Structure of cryptochromes. Cryptochromes are flavoproteins, which means they contain a flavonoid mononucleotide FMN as a cofactor essential for their light-sensing activity. The structure of cryptochromes is composed of a central chromophore binding domain, responsible for the light-absorbing properties, and AC terminal domain, which is involved in signaling and interaction with other proteins. The FMN cofactor undergoes a photoisomerization reaction upon exposure to blue light, leading to a conformational change in the protein that activates its signaling pathway. This structural transformation enables cryptochromes to interact with downstream signaling components to regulate various biological processes. Cryptochromes are distinct from phototropins and other class of blue light receptors in their function and the light wavelengths they sense. While phototropins primarily mediate phototropism, the directional growth response to light, cryptochromes are involved in circadian rhythm regulation and light-induced gene expression. Function of cryptochromes. Cryptochromes are involved in a variety of crucial processes in plants, including circadian rhythm synchronization, seedling development, photomorphogenesis, and flowering regulation. Their main functions include circadian rhythm regulation. Cryptochromes are key components of the plant's circadian clock, which helps synchronize internal biological processes with the external light-dark cycle. By sensing blue light, cryptochromes influence the expression of clock genes, ensuring that the plant's daily physiological processes, like stomatal opening, photosynthesis, and growth patterns, are aligned with the day-night cycle. Cryptochromes modulate the expression of timing of CAV1, TOP1, and gigantea GI, which are important regulators of the circadian clock. Photomorphogenesis and seedling development. In the absence of light, seeds and seedlings exhibit etiolated growth, characterized by long stems and underdeveloped leaves. When exposed to light, cryptochromes play a key role in promoting detylation, a process in which the seedling stops detylation, begins chloroplast development, and starts leaf expansion. Cryptochromes help mediate these responses through the regulation of light-responsive genes, optimizing the plant's ability to transition from a dark to a light environment. Regulation of flowering time. Cryptochromes are involved in the regulation of flowering in response to light, particularly in the regulation of photoperiodic flowering. In some plants, the conversion of cryptochromes to an active form in response to blue light helps integrate environmental light cues with the plant's internal developmental clock. This allows the plant to flower at the appropriate time, ensuring reproductive success. Phototrophism and directional growth. While cryptochromes are not the main photoreceptors for phototrophism, a process mediated by phototropins, they can still influence the directional growth of plants under blue light. Cryptochromes contribute to shade avoidance and light optimization, helping plants adjust their growth patterns in response to light gradients, particularly in the early stages of seedling development. Mechanisms of action of cryptochromes. The molecular mechanism of cryptochrome action to blue initiate photomorphogenesis. Answer: COP1 constitutive PHOTOMORPHOGNIC1 is degraded by cryptochrome action in response to blue light, initiating photomorphogenesis. CSIR Net Deck 2018. Cryptochromes in plants primarily respond to which type of light? Answer: Cryptochromes primarily respond to blue light and influence various developmental processes like photomorphogenesis and circadian rhythm regulation. Conclusion. Cryptochromes are fundamental to light-mediated developmental processes in plants, influencing circadian rhythms, photomorphogenesis, and flowering in response to blue light. Through their structural properties and activation mechanisms, cryptochromes act as blue light sensors that activate signaling cascades leading to changes in gene expression and physiological responses. Understanding their functional roles and molecular mechanisms provides critical insights into plant growth and adaptation to changing light environments. Would you like to continue with another section of sensory photobiology? You said, phototropins, ChatGPT said. Phototropins, structure, function, and mechanisms of action. Phototropins are a class of blue light receptors that play a critical role in regulating various plant responses to light, including phototrophism, stomatal opening, and chloroplast movement. These receptors are essential for the plant's ability to sense and adapt to light conditions, which is crucial for optimal growth and development. Phototropins are primarily involved in mediating plant responses to blue light around 400 to 500 nm, and they work in concert with other photoreceptors like cryptochromes and phytochromes to modulate various physiological processes. Structure of phototropins. Phototropins are flavotropins, containing flavin mononucleotide FMN as a chromophore. The structure of phototropins includes photoreceptor domain, love domain. The light, oxygen, or voltage love domain is responsible for the light sensibility of phototropins. This domain binds to the FMN chromophore, and upon exposure to blue light, the chromophore undergoes photoisomerization, leading to a conformational change in the protein structure. This change activates downstream signaling pathways. Kinase domain. Phototropins also contain a serine threonine kinase domain that is crucial for their signaling function. The kinase domain is involved in phosphorylation events that activate or deactivate various downstream proteins involved in light-mediated responses. C-terminal domain. The C-terminal domain is involved in protein-protein interactions and the regulation of cellular processes like chloroplast movement and stomatal opening. Function of phototropins. Phototropins are involved in regulating several important processes in plants, most notably phototrophism, stomatal opening, and chloroplast movement. These processes ensure that plants optimize their ability to capture light for photosynthesis and adapt to changes in their environment. 1. Phototrophism. Phototrophism is the directional growth of plants in response to light. Positive phototrophism occurs when plants grow toward a light source, while negative phototrophism occurs when plants grow away from light. Phototropins mediate positive phototrophism, enabling the plant to grow toward light, especially in low light conditions, ensuring optimal photosynthesis. This response is crucial for seedlings, which use phototrophism to orient themselves toward light sources. Mechanism. Upon blue light exposure, the phototropin photoreceptor undergoes a conformational change in the love domain, which activates its kinase activity. The kinase domain phosphorylates target proteins, leading to asymmetric growth of the plant on the shaded side of the stem. This growth results in the bending of the stem toward the light source. 2. Stomatal opening. Phototropins play a key role in regulating stomatal movement, which is essential for controlling gas exchange, CO2, and water loss through transpiration. In response to blue light, phototropins mediate the opening of stomata to facilitate CO uptake for photosynthesis. Mechanism. In response to blue light, phototropins are activated and trigger a signaling cascade that leads to the activation of ion channels in guard cells surrounding the stomata. The movement of ions like K+ into the guard cells causes water to enter, resulting in turgor pressure and opening of the stomatal pore. This process enables gas exchange and transpiration regulation. 3. Chloroplast movement. Phototropins are involved in the movement of chloroplasts within plant cells in response to light intensity. This process is crucial for optimizing light absorption while protecting the plant from excessive light that could damage cellular structures. Mechanism. Under low light conditions, chloroplasts move toward the light source to maximize light absorption for photosynthesis. In high light conditions, chloroplasts move away from the light source to minimize photodamage caused by excessive light. Phototropins mediate this movement by activating downstream signaling pathways that control the motor proteins responsible for chloroplast relocation within the cell. 4. Leaf expansion. Phototropins are involved in leaf expansion in response to blue light. This process is crucial for maximizing photosynthetic surface area. The presence of blue light triggers the expansion of leaves, which increases the plant's ability to capture light for energy production. Mechanism. Phototropin activation triggers a series of molecular events that lead to the growth of leaf cells and the subsequent expansion of the leaf. This process helps the plant increase its surface area for photosynthesis and adapt to environmental light conditions. Mechanisms of action of phototropins. The primary mechanism by which phototropins function involves their ability to sense blue light and trigger signal transduction pathways that regulate plant growth and development. Photoisomerization and conformational change. Upon exposure to blue light, the FMN chromophore in the phototropin love domain undergoes photoisomerization, resulting in a change in the conformation of the protein. This conformational change activates the kinase domain, which becomes enzymatically active and can phosphorylate target proteins involved in plant growth responses. Phosphorylation and signal transduction. The kinase domain of phototropins mediates phosphorylation of downstream signaling components, such as PIN-FOR and MED, PIN proteins involved in oxygen transport. The phosphorylation of these proteins leads to polar oxygen transport, which is essential for phototrophism and other growth responses. Activation of ion channels for stomatal movement. Phototropin activation also influences ion channels in guard cells. For example, the phosphorylation of specific proteins in guard cells leads to the activation of potassium K+ channels, which causes the influx of K+ ions. This ion influx leads to water movement into the guard cells, resulting in stomatal opening. Coordination with other photoreceptors. Phototropins often work in tandem with other photoreceptors, such as cryptochromes and phytochromes, to regulate growth processes like photomorphogenesis and flowering. While cryptochromes mediate blue light responses related to circadian rhythms, phototropins are primarily involved in directional growth responses such as phototrophism and chloroplast movement. Role of phototropins in plant growth and development. Phototropins are essential for the plant's ability to adapt to environmental light conditions, ensuring optimal light capture and photosynthesis while minimizing damage from excessive light. Their roles in phototrophism, stomatal regulation, and chloroplast movement are crucial for maximizing plant fitness and survival in a constantly changing environment. Phototropins also contribute to photomorphogenesis, the light-dependent developmental process that ensures the proper formation of leaves, stems, and flowers. By coordinating light signals with growth responses, phototropins help plants optimize their energy production and reproductive success. Previous year CSIR Net questions. CSIR Net Deck 2020. Which type of light does phototropin primarily respond to, and what is its main role in plants? Answer. Phototropins primarily respond to blue light and mediate phototrophism, stomatal opening, and chloroplast movement. CSIR Net June 2019. What is the primary signaling mechanism triggered by phototropin activation in plants? Answer. Phototropin activation results in the phosphorylation of target proteins, leading to asymmetric growth during phototrophism and ion channel activation for stomatal opening. CSIR Net Deck 2018. What is the role of phototropins in leaf movement and expansion? Answer. Phototropins regulate leaf expansion in response to blue light by promoting cell growth and optimizing light absorption for photosynthesis. Conclusion. Phototropins are key blue light receptors in plants, mediating important processes such as phototrophism, stomatal regulation, and chloroplast movement. Their ability to sense and respond to light enables plants to adapt to their environment, optimize photosynthesis, and regulate their growth and development. The activation of phototropins triggers signaling cascades that affect various physiological responses, making them essential for maintaining plant fitness and survival. Would you like to continue with another section or topic? You said, stomatal movement. ChatGPT said. Stomatal movement, mechanisms and regulation in plants. Stomatal movement refers to the opening and closing of the stomata, which are microscopic pores present on the surfaces of plant leaves and stems. Stomatal movement is crucial for regulating gas exchange, allowing CO to enter the plant for photosynthesis while enabling oxygen, O, and water vapor to exit during transpiration. The movement of stomata is controlled by several environmental, internal, and hormonal signals to ensure optimal water use efficiency and carbon fixation. Structure of stomata. Stomata are made up of two guard cells that surround the central pore. These guard cells are highly specialized and can change their shape to open or close the pore. The shape changes occur due to the osmotic changes in the guard cells, which are regulated by the influx or efflux of ions, particularly potassium ions, K+, chloride ions, Cl, and malate. Guard cells. The guard cells contain specialized chloroplasts and are responsible for the light-dependent regulation of stomatal opening. They are asymmetric in shape, with the inner walls thicker than the outer walls. This structure allows for changes in their turgor pressure, leading to stomatal movement. Stomatal pore. The stomatal pore is the opening between the two guard cells, and its size determines the rate of gas exchange and water loss. The size of the pore is dynamically regulated by the guard cells in response to environmental conditions such as light intensity, humidity, CO concentration, and water availability. Mechanism of stomatal opening. Stomatal opening is driven primarily by turgor pressure in the guard cells. The process involves the active transport of ions into the guard cells, followed by water movement, which increases the turgor pressure and causes the guard cells to swell, thus opening the stomatal pore. Blue light activation. The photoreceptor phototropins, which respond to blue light, are crucial for stomatal opening. Upon blue light exposure, phototropins activate a signaling pathway that leads to the influx of K+ ions into the guard cells. This increase in ion concentration inside the guard cells causes water to enter through osmosis, increasing turgor pressure and resulting in stomatal opening. Proton pump activation. In response to blue light, the H+ Ap proton pump is activated in the guard cell membranes. This pump actively transports protons H+ out of the guard cells, creating an electrochemical gradient that favors the influx of potassium K+ and chloride Cl ions. The accumulation of these ions inside the guard cells reduces the water potential, drawing water into the cells by osmosis, causing the guard cells to become turgid and opening the stomatal pore. Role of auxin. Auxin, a plant hormone, also plays a role in stomatal opening. It can stimulate ion channel activation and influence the movement of ions into the guard cells, contributing to stomatal aperture regulation. Role of abscisic acid ABA. Abscisic acid ABA, a plant hormone, is involved in stomatal closure, especially under stress conditions like drought. ABA promotes the release of calcium ions Ca², which activate protein kinases that disorder the ion channels, leading to the efflux of K+ and Cl in the guard cells. This reduction in ion concentration causes water to exit the cells, leading to a decrease in turgor pressure and the closing of the stomatal pore. Mechanism of stomatal closure. Stomatal closure is primarily regulated by the plant's need to conserve water, especially under conditions of water stress or high temperatures. ABA-mediated closure. When a plant experiences drought stress, ABA is synthesized and transported to the guard cells, where it triggers the closure of stomata. ABA induces the activation of calcium Ca², signaling pathways and the activation of protein kinases. This results in the efflux of K+ and Cl ions from the guard cells, leading to a decrease in turgor pressure, causing the stomatal pore to close. Environmental signals. Stomatal closure is also regulated by high temperatures, high light intensity, and low humidity. These environmental factors induce ABA synthesis and other signaling molecules that coordinate the closure of stomata to prevent water loss through transpiration under stressful conditions. Calcium signaling. Calcium Ca² plays a critical role in mediating stomatal closure. The increase in cytosolic Ca² levels, triggered by ABA stress signals, activates various protein kinases and ion channels that cause the loss of K+ ions from the guard cells. This reduces the guard cell's turgor pressure, resulting in stomatal closure. Regulation of stomatal movement by environmental factors. Stomatal movement is highly responsive to changes in the plant's environment. Several external factors influence whether stomata open or close, optimizing the plant's photosynthetic activity and water use efficiency. Light. Blue light is the most important environmental cue for stomatal opening, as it activates phototropins in the guard cells. However, red light and white light can also trigger stomatal opening, although blue light is more potent in this regard. CO concentration. High levels of carbon dioxide (CO) inside the leaf lead to stomatal closure. This response is a mechanism to conserve water when there is sufficient CO available for photosynthesis. Conversely, low CO levels lead to stomatal opening, allowing for more CO to enter for photosynthesis. Humidity. Low humidity causes water loss through transpiration, triggering stomatal closure to conserve water. Conversely, high humidity reduces the rate of water loss, promoting stomatal opening to facilitate gas exchange. Temperature. High temperatures increase transpiration rates and can induce stomatal closure to minimize water loss. However, very high temperatures may also cause stomatal opening as part of the plant's heat tolerance mechanisms. Signaling pathways and hormonal regulation. The regulation of stomatal movement involves a complex network of signaling molecules, hormones, and environmental signals. The key hormones involved include abscisic acid ABA. ABA is the primary hormone responsible for stomatal closure under water stress and high temperature conditions. Auxin. Auxin helps in the opening of stomata by influencing the movement of ions into the guard cells. Gibberellins Ca. Gibberellins promote stomatal opening by stimulating ion transport into the guard cells. Cytokinins. Cytokinins can influence stomatal closure by interacting with ABA signaling pathways. Previous year CSIR net questions. CSIR net June 2020. What is the primary role of abscisic acid ABA in stomatal movement? Answer. ABA primarily regulates stomatal closure under stress conditions, such as drought, by activating calcium signaling and inducing the efflux of ions from guard cells. CSIR net 2018. Which light is most effective in triggering stomatal opening? Answer. Blue light is the most effective in activating phototropins, which in turn triggers stomatal opening. CSIR net June 2019. Explain the mechanism of stomatal opening in response to blue light. Answer. Blue light activates phototropins, which stimulate the H+ Ap proton pump, leading to the influx of K+ ions into the guard cells, causing water to enter by osmosis, increasing turgor pressure, and resulting in stomatal opening. Conclusion. Stomatal movement is a complex and finely regulated process that allows plants to balance gas exchange and water conservation. It is influenced by various environmental cues, such as light, CO concentration, temperature, and humidity, and regulated by hormones like ABA, auxins, and gibberellins. The ability of plants to control stomatal aperture optimally is essential for maintaining photosynthesis, transpiration, and overall water use efficiency. You said. Photoperiodism. ChatGPT said. Photoperiodism in plants. Photoperiodism refers to the physiological response of plants to the relative lengths of day and night. It plays a crucial role in regulating various developmental processes in plants, such as flowering, germination, bud formation, and vegetative growth. Photoperiodism helps plants determine the right time for key developmental events based on the timing of the seasons, ensuring their survival and reproduction in optimal environmental conditions. Types of photoperiodic responses. Plants can be categorized based on their response to day length into three main types: short day (SD), long day (LD), and day neutral (DN) plants. Short day plants (SDPs). These plants require a long night more than a critical duration and short day to flower. They typically flower when the night duration exceeds a critical threshold. Examples: chrysanthemum, soybean, and poinsettia. Mechanism: the long nights are perceived by the plant, and this triggers the production of florigen, the flowering hormone, leading to flowering. Long day plants (LDPs). These plants require a short night less than a critical duration and long day to flower. They typically flower when the day length exceeds a critical threshold. Examples: spinach, lettuce, and wheat. Mechanism: a shorter night period is critical for flowering, which is induced by the production of florigen in response to extended daylight. Day neutral plants (DNPs). These plants are not significantly affected by day length and can flower under a wide range of light conditions. Examples: tomato, cucumber, and corn. Mechanism: the flowering of these plants is controlled by other environmental factors, such as temperature, rather than photoperiod. Photoperiodic induction of flowering. The process of photoperiodic induction of flowering involves the perception of light signals by specialized photoreceptors in the plant. The key factors in this process include phytochromes. Phytochromes are light-sensitive proteins that perceive the red and far red light spectrum. They play a critical role in the plant's ability to measure day length. In long day plants, a high ratio of red light PR to far red light PFR during the day helps the plant determine the length of the day. Cryptochromes and phototropins. These are other photoreceptors involved in blue light perception. They also help regulate photoperiodic responses, especially in plants that require blue light for germination or other growth stages. The role of the circadian clock. The circadian clock in plants controls their daily rhythms, which include the timing of gene expression in response to light. The photoperiodic signal is integrated with the plant's circadian rhythm to synchronize flowering with the optimal time of year. The gene constant CO is a central component of this process, and it is activated by light. CO promotes the expression of flowering locus T, FT, the florigen, which is transported to the shoot apex and triggers flowering. The role of florigen, FT. Florigen is a flowering hormone that plays a central role in photoperiodic induction of flowering. When the plant perceives the photoperiod correctly, the FT gene is activated, and the FT protein is synthesized. This protein moves from the leaves to the shoot apical meristem, where it induces flowering. Mechanism of photoperiodism. The photoperiodic process begins when the plant perceives the duration of light and dark periods, which is facilitated by photoreceptors like phytochromes and cryptochromes. The plant then integrates this information with its internal circadian clock, which operates on a roughly 24-hour cycle, ensuring synchronization with the natural light cycle. Light signal perception. Red light 660-680 nanometers is perceived by phytochrome in the PR state, which absorbs red light. This causes a conformational change to the PFR state, the active form. The balance between PR and PFR states helps the plant track day and night cycles. Far red light 730-740 nanometers also affects the phytochrome system, with PFR being converted back to PR. The ratio of PFR to PR determines the photoperiodic response. Circadian clock and gene expression. Plants have a circadian clock that regulates the expression of genes involved in flowering. Constant CO gene expression is regulated by light, and CO protein is stabilized by light during the day. CO activates the expression of flowering locus T, FT, which is the florigen. Transport of florigen. The FT protein moves from the leaf to the apex of the shoot, where it activates flowering pathways and triggers the transition from vegetative to reproductive development. Photoperiodic control of flowering. Short-day plants require a long night for the accumulation of the FT protein. This enables the activation of the flowering program. Long-day plants require a longer period of daylight to accumulate enough PFR and activate Colorado, which then leads to the expression of FT and flowering. Factors affecting photoperiodism: light quality. The quality of light, particularly the ratio of red to far red light, influences the plant's response to photoperiods. Plants sensitive to light quality will respond based on whether they perceive more red or far red light. Temperature. Temperature interacts with photoperiodism, often acting synergistically with light. For instance, some plants require both the correct photoperiod and a certain temperature for flowering to occur. Vernization. Some plants, especially winter annuals, require a period of cold temperatures vernalization before they can flower, even if the correct photoperiod is present. Light intensity. Light intensity, though not directly involved in photoperiodic control, can influence the overall health of the plant and its ability to perceive photoperiod signals effectively. Photoperiodism and agricultural practices. Photoperiodism has significant implications for agriculture. Understanding the photoperiodic requirements of plants is essential for crop management. In agriculture, manipulating flowering time. By controlling light exposure, farmers can manage flowering time in crops like rice, wheat, and soybean to maximize yield and synchronize harvests. Season extension. In areas with short growing seasons, artificial light can be used to extend the growing period of long day plants or delay the flowering of short day plants. Indoor and controlled environments. Growing plants under controlled indoor conditions, e.g., greenhouses, often involves manipulating photoperiod using LED lighting to simulate specific day-night cycles, optimizing flowering and fruiting. Previous year CSIR net questions. CSIR net June 2019, which of the following is most likely to influence photoperiodism in plants? Answer: light quality, temperature, and photoperiod are the major factors influencing photoperiodism. CSIR net 2018, in long day plants, the critical factor for flowering is the duration of night. Which of the following statements is true regarding photoperiodism in long day plants? Answer: long day plants require short nights and long days for flowering, and the response is mediated by the photoreceptors that detect day length. CSIR net June 2017, which hormone plays a central role in regulating flowering in response to photoperiodism? Answer: florigen-4 is a central hormone responsible for photoperiodic induction of flowering in plants. Conclusion. Photoperiodism is a crucial mechanism for plants to synchronize their growth and reproductive cycles with environmental conditions, ensuring their survival and reproductive success. It enables plants to time their flowering with optimal conditions for pollination and seed production. The photoperiodic response is mediated by light receptors, circadian rhythms, and the production of florigen, which is essential for flowering. Understanding photoperiodism has vast applications in agriculture, from crop yield optimization to controlled environment agriculture. You said biological clocks. ChatGPT said. You said biological clocks. ChatGPT said biological clocks in plants. A biological clock is an internal mechanism that allows organisms to anticipate and adapt to regular environmental changes, such as the day-night cycle, seasonal variations, and temperature fluctuations. These clocks are essential for coordinating various biological processes with external rhythms, ensuring optimal timing for activities like germination, flowering, fruiting, and photosynthesis. In plants, the biological clock is most commonly referred to as the circadian clock, and it regulates many physiological processes in a 24-hour cycle, synchronized with the Earth's rotation. The plant's biological clock is influenced by external cues, primarily light and temperature, and ensures that various processes occur at the right time during the day or night. The circadian rhythm in plants. Circadian rhythms are roughly 24-hour cycles of physiological processes, including gene expression, metabolic activity, and behavioral changes. In plants, these rhythms are synchronized with the light-dark cycle, ensuring that plants perform key functions at the most advantageous times for survival and reproduction. The circadian rhythm influences several aspects of plant life, including leaf movement, opening and closing of stomata or leaf blades, photosynthesis, timing of photosynthetic processes, flowering, regulation of flowering time in response to environmental cues, growth patterns, coordination of cell division and elongation, hormonal regulation, influencing the synthesis and action of plant hormones like gibberellins, auxins, and cytokinins. Components of the biological clock in plants. The biological clock in plants operates through an intricate system of genes and proteins that interact with external signals. The major components include clock genes, clock or CCA1 in Arabidopsis. These are genes that play a central role in maintaining the plant's 24-hour rhythm. TOC1, timing of CAB1, involved in regulating the output of the clock to affect light responsive genes. LHY, late elongated hypocodal, a gene that contributes to the regulation of the plant's response to light. PRR, pseudo-response regulator genes. These genes regulate the expression of other genes involved in the circadian cycle. Photoreceptors. Phytochromes, perceive red and far red light, playing a role in regulating circadian rhythms and photoperiodism. Cryptochromes, sensitive to blue light and also involved in the regulation of the biological clock, particularly in short day or long day plants. Phototropins, control circadian rhythms related to light perception, mainly involved in blue light sensing. Proteins and transcription factors. The clock genes produce proteins that bind to DNA and regulate the expression of other genes according to the time of day. These proteins typically form feedback loops, where their expression is both regulated by and regulates their own production. The oscillator. The plant circadian clock is based on feedback loops formed by transcription factors and their target genes. For instance, the expression of LHY and CCA1 proteins regulates the expression of the top 1 gene, and top 1 in turn regulates LHY and CCA1, forming a negative feedback loop. Mechanisms of biological clocks in plants. The plant circadian clock operates on a 24-hour cycle by using feedback loops to regulate gene expression in a rhythmic pattern. These loops ensure that processes like photosynthesis, growth, and flowering are coordinated with the light-dark cycles. Here's how it works. Morning phase light activation. During the morning light phase, light stimulates the expression of certain genes, such as LHY and CCA1, which activate genes involved in photosynthesis and metabolism. These genes prepare the plant for the day's activities, including stomatal opening, growth, and energy production through photosynthesis. Midday phase peak of activity. At midday, the plant's metabolic processes are most active, with peak photosynthesis and nutrient uptake. The PRR genes help fine-tune the plant's response to external cues, ensuring peak productivity. Evening phase gene silencing. During the night dark phase, the plant begins to wind down its activities. The expression of LHY and CCA1 decreases, while top 1 increases. This shift in gene expression helps regulate processes such as energy storage, e.g., starch conversion, and prepares the plant for the next life cycle. Night phase gene repression and reset. In the night phase, the plant's activity level decreases. The repression of genes involved in photosynthesis and active growth is important for ensuring energy conservation. This phase also involves resetting the biological clock for the next life cycle, ensuring that the plant remains synchronized with the external 24-hour light-dark cycle. Importance of biological clocks in plants coordination of growth and development. The circadian clock helps plants coordinate their growth processes to occur at optimal times. For example, leaf movements or changes in stomatal aperture are controlled by the circadian clock to maximize photosynthesis during daylight and minimize water loss during the night. Energy efficiency. By regulating the timing of photosynthesis and respiration, the plant ensures efficient energy use. The circadian clock helps synchronize chloroplast activity, ensuring that energy production aligns with the availability of light. Reproductive timing. Biological clocks regulate processes like flowering and fruit ripening. In some species, the clock helps ensure that these processes occur at the right time in relation to environmental conditions, such as the length of the growing season or temperature. Response to environmental stress. Plants can sense environmental changes, e.g., temperature shifts, light intensity, and use the biological clock to adjust their physiological responses. This enables plants to anticipate and adapt to environmental stresses, such as drought or extreme temperatures, to enhance their survival. Plant-human interaction. The knowledge of plant biological clocks has implications for agriculture and horticulture. Manipulating the circadian rhythms of crops can optimize growth and flowering times, improve yield, and make crops more resilient to environmental stresses. Key components of biological clocks in plants. Circadian oscillator consists of genes like LHY, CCA1, and TOP1, which produce proteins that regulate the expression of other genes. Light and temperature signals. Light and temperature are the two primary external signals that synchronize the plant's biological clock. Light affects the timing of gene expression, while temperature provides additional fine-tuning. Regulation of gene expression. Proteins encoded by clock genes act as transcription factors to regulate the expression of downstream genes that control growth, metabolism, and other plant functions. Previous year CSIR net questions. CSIR net June 2018. What is the central role of the biological clock in plants? Answer. The biological clock in plants regulates daily rhythms like photosynthesis, growth, and flowering in response to light and dark cycles. CSIR net 2017. Which component is most responsible for maintaining circadian rhythms in plants? Answer. Clock genes such as LHY, CCA1, and TOP1 are responsible for maintaining circadian rhythms in plants. CSIR net June 2016. The circadian clock in plants is primarily synchronized by which environmental factor? Answer. Light, specifically its red and blue components, is the primary environmental factor that synchronizes the plant's circadian clock. Conclusion. Biological clocks, particularly circadian rhythms, play a critical role in regulating various physiological and developmental processes in plants. By synchronizing activities such as photosynthesis, flowering, and growth with the external light-dark cycle, these clocks enhance the plant's efficiency and adaptability. The molecular mechanisms behind the biological clock involve complex interactions between genes, proteins, and photoreceptors, which ensure that plants function optimally in response to environmental cues. Understanding the biological clock offers valuable insights into enhancing agricultural practices, improving crop productivity, and increasing plant resilience to environmental stress. Search.